Yeah, thank you very much for allowing me to present my work here. The font got a bit more edgy, but I hope you can still read everything. So it's I'm going to talk about distance and hierarchy-dependent functional disconnectivity in schizophrenia, another schizophrenia talk. I have to say this is going to be a pretty straightforward neuroimaging study. And I also should tell you that this is a real new avenue for me, the whole computational psychiatry, as I spent the last like 13 years working mostly on models of consciousness. But now it's time for a bit more down to earth actual neuroscience. And it's a bit daunting to step into this field of computational psychiatry, because if you do a PubMed search on schizophrenia and MRI, there are 11,000 papers, right? And all nice complex analyses that have been proposed have been applied to existent data, right? So nevertheless, there's still this question about how the structural and functional alterations match. And so even though we have associated schizophrenia with a widespread functional disconnectivity for years, the spatial scale and the structural correlates of these alterations still remain unclear to some extent, right? So in other words, the structural deficits and the functional deficits don't always match up, and we're trying to think about a bit what could underlie these reasons. And one idea that we had is that the short-range cortical connections are really difficult to assess with our standard diffusion imaging tools, and they may therefore be systematically underappreciated. So you can see here the DTI tractography. It mostly captures the white matter connections between regions, right? But not the local connectivity within a region. And the same is true for our standard parcellation-based functional connectivity analyses, where you look between regions, but of course you're not accessing what happens within the region. And that's not homogeneous. So for all we know, and we don't know very much actually about local structural connections and their organizations in the cortex, but there is a difference, and there's a gradient between sort of the more posterior sensory regions and the more frontal multimodal regions, where the sensory regions are mostly organized in a topographical manner, in a grid-like manner with a lot of local lateral connectivity. Whereas the more frontal regions, they have a much lower percentage of inputs coming from local lateral connections, right? And so this motivated us to hypothesize that maybe if we look at short-range functional connectivity, we can match that up a little better with microstructural abnormalities, and that there's also potentially a relation between the functional and structural deficits and cortical hierarchy. So this is what we aimed to test, and we used two public data sets from the Schizconnect website, the COBRA and the NMORPH data set, with 86 schizophrenia patients and 99 healthy controls. So what did we do for the distance-dependent functional connectivity strength? Straightforward, we took the bold time series, converted that from the voxel space into surface space, computed the functional connectivity of all the surface vertices with each other. And we also computed the geodesic distance, so blowing up the cortical surface into a ball. And then you can just analyze how far away on that surface is every surface vertex from every other surface vertex. And then combining these two, we computed a functional connectivity strength that takes the average functional connectivity of a vertex with all the other vertices in a specific radial distance, right? So you take voxel vertex here, for example, this little patch, and you look at all the other ones that are between 5 and 15 millimeters away from it and compute the average functional connectivity strength, mean FC for that voxel, and you get a map of all the vertices and how strong they're connected to other vertices in that. And you can see here that this reflects what I told you earlier, that there is a more short range, stronger connectivity in the more posterior sensory regions than in the more frontal regions. Now, the final step is to contrast this in healthy controls and the schizophrenia patients. So we did that for a range of distances, and we did find some significant clusters, namely in short range connectivity, we found deficits in the bilateral dorsal somatomotor, somatosensory cortices. And then complementary, we found an increase in functional connectivity strength at longer range distances in the precuneus. And the important part here is that this short range deficit was not reflected in the overall global functional connectivity strength. So you would only capture that if you actually do the distance dependent analysis, not if you do the overall global functional connectivity strength, whereas the longer range connectivity was reflected in the overall connection. So we're also not the first ones to do a distance dependent analysis, but previous papers have just looked at mostly under 70 millimeters and over 70 millimeters, and also mostly used just a Euclidean distance between voxels, which has a lot of problems with like going from one hemisphere to the other. You capture things that aren't actually connected, right? So we did the surface analysis here. All right, so we also looked at just the overall distribution of our differences between the patients and the controls. And looking at that a little bit, the pattern might look familiar in that we found increases in what is default mode network regions, for example, and we found decreases mostly in sort of the sensory areas. And so there was this very nice paper by Margulies in 2016 that analyzed gradients in the functional connectivity from the human connectome data. And they found a primary gradient that goes along the axis of more primary unimodal areas to more multimodal or transmodal, as we call them here, areas. And so we took their assignment of these resting state networks into these three classes, the primary, intermediate, and transmodal, and averaged the vertices here into these three classes of a hierarchy. And we did the same distance dependent analysis for that, and found that now if you do the average measures, you see that for all the distance ranges, you find decreased connectivity in the more primary location, like cortical areas, whereas you find a increase in connectivity in the more transmodal areas. Now, you might have noted that there is this little R in front of the FCS, the functional connectivity strength, and that stands for relative functional connectivity strength, because what we did here for that analysis and also the cluster analysis I showed you before is that we z-scored within subjects, because we have two data sets with two different scanners, and we tried to make this all a bit more consistent. Now, we also did the analysis just taking the absolute functional connectivity scores, and in those absolute values, you still see the decrease at more shorter ranges in the patients, but the increase in the higher modal, the transmodal areas is not significant. And I want to highlight this because this also might contribute a bit to the diverse findings in the literature, that it really does depend on the details of the analysis, especially when you do things like averaging or cluster analysis. Because actually, if you just compare the the z-score maps, they look very similar between the relative and the absolute analysis. Just the results with thresholding and whatever may look different. And nevertheless, this is also maybe a bit of an indicator that the findings for the higher area might be driven by compensatory to the deficits in the more primary areas, right? So the idea would be that maybe you have this deficit in primary areas that can be compensated to some degree, and then leads to a relative increase in the longer range connectivity that is often observed in a regional analysis. All right, so the correspondence with microstructure. How do you even assess cortical microstructure? There isn't much you can do non-invasively, but there are two proxies that we use to assess intracortical microstructure. One is a well-known T1-T2 ratio that was introduced to capture myelin, and it has been criticized that it doesn't actually capture myelin that well, but it is still a useful indicator of sort of microstructural integrity. So that is the one measure we used. And then we also introduced a sort of principled way of doing a functional connectivity density, where what that is you take the functional connectivity, choose a threshold, and binarize around that threshold. So everything that is higher than, let's say, 0.4, you count as a true connection, and everything lower than 0.4 is not a true connection. Then you can count those, the degree for that. And usually this threshold is just, you know, arbitrarily chosen in a way. But what we did is to think about how we capture actual anatomical connections. And to do that, we chose two regions that are known to not be anatomically connected, namely the cuneus and the contralateral medial frontal gyrus. So any correlations between these areas are not driven by direct anatomical connections. And we looked at the distribution of functional connectivity values for each individual subject. Here you can see one in blue, and compare that with the red distribution is within the cuneus, so local and anatomically connected areas, which are higher. And then we took a very conservative threshold that is just the maximum value of correlation between these unconnected regions, and use that as the threshold. So what we get as a result is that everything above that threshold is most likely a true anatomical connection, right? But we're also missing a lot of actual anatomical collections. So the ones we have are true positives, but we're missing a lot of other anatomical connections. When we do this analysis, we also do find a significant cluster for this functional connectivity density in the same region of the dorsal somatosensory cortex. On the right side, it was there on the left side too, but not significant. And here, so the T1, T2 clusters all did not reach significance, but we still looked at the overlap between all these non-significant clusters that we, not significant after correction. So we looked at all these clusters, and the only region where they all overlap, so the three measures, the two microstructural measures and the functional connectivity strength overlap in this somatosensory area. So this is some confirmation that the functional connectivity deficits we see here actually come in a straightforward way from missing like anatomical connectivity strength here. You can do the same correlation now with the hierarchical functional connectivity averages. And we also find that the correlation between the anatomical measures and the functional connectivity measures is highest at lower distances and for the primary networks. So to simplify, there's a good correspondence between the structural deficits and the functional deficits in the shorter range connectivity and for the more primary areas. Okay, we also did a correlation with psychiatric symptoms. The only the COBRA data set has the psychiatric scores, so we had fewer subjects there, and so there's nothing here reached significance after correction, but I still want to show you the trend here is that the cluster, the dorsal somatosensory cluster we identified, does correlate, although not significantly after correction, with negative symptoms on the right side, and then on the left side, the homolog on the left side correlated with age, and that makes sense, as also Andrea mentioned, that there is this accelerated aging in schizophrenia patients. So the correlation with age is interesting here. And so I'm going to cut this short, but the general trend is also with now the hierarchical analysis that there's sort of an opposite correlation between the primary networks and the transmodal networks with respect to their correlation with the positive and the negative symptoms, right? So even though not much here is significant, you can see that the trend just goes in the opposite direction. All right. So the conclusion of this work is that our findings reveal two distinct patterns of cortical disconnectivity. One is the short range reductions in primary sensory cortices, and that co-localizes with the microstructural abnormalities that we measured. And then we have the longer range relative increases in the transmodal regions, and they are structurally decoupled at the local level, right? So that doesn't mean that there aren't structural deficits in the white matter tracts. We didn't evaluate those, right? But at the local level, they're not represented. So this distance and hierarchy-dependent framework highlights that there is local disconnectivity, which is complementary to the more conventional region-based or diffusion-based approaches. And just as an outlook on how this connects to some other findings in schizophrenia and future work that we want to do, there's also evidence now that the spindles, so the sleep spindles, the EEG signature, is reduced in schizophrenia patients. You can see that here there's a very nice study, and now extension of that study by Kozemiakov and Elife. So yes, so there's an overall reduction in the density, amplitude, and so on in schizophrenia patients. And that so sleep spindles are a biomarker. And there was a very nice paper by Baran et al. That showed that in this somatosensory region that we identified, first of all, they found that there is a thalamocortical increased functional connectivity in schizophrenia patients, whereas we found the reduction in the cortical-cortical, right? So, and that may go together in that the increase of the thalamocortical may lead to a decrease in the cortical-cortical connectivity. And also it is this region too that both in healthy subjects and schizophrenia patients correlates with the amount of spindles that the subjects have, right? So hopefully we can bring this all together, and we're planning to do that maybe with modeling and also with non-invasive neurostimulation experiments, trying to find this connection of thalamocortical with cortical deficits in the schizophrenia patients. Thank you very much.
I think there's time for